This is the last section of chapter 12 that I'll review, and then you should have been working on chapter 13 and that associated parts, and there's another video for that. So kind of the last section, last 30 or so slides that we're going to go over is the last half of chapter 12, and this is how neurons communicate with each other. We'll start with A, ways they communicate first, and then we'll kind of move on to a few other things. So when we have groups of neurons that are communicating with each other, oftentimes they can do a number of different actions, which we call types of circuits. And the two that we focus on in this course are convergence and divergence. Also, you could just refer to them as converging and diverging. And this is kind of explaining how groups of neurons are communicating with each other. And we'll kind of see a picture of what that looks like. This type of communication could be localized, meaning it's located and whatever function is happening is in the same area. So those groups of neurons and whatever function or effector we're trying to work with is in the same area. Or it could be distributed to over a larger area where we have to communicate over a longer area of space. So we'll look at convergence first, and it is exactly what you would think it would be if we were all converging on one person, okay, or one neuron. This is when a single neuron receives input from multiple neurons. You can see in this picture here, we have groups of multipolar neurons, okay? And each of these is sending a signal through their axon, and it's being collected by the dendrites of this receiving one, okay? They're all converging on that one neuron. What this can do is help kind of intensify a signal. It can allow us to receive kind of complex signals. Okay, so we're getting signals from multiple neurons converging onto one, and it has to process all of those signals. We see this for sight, sound, smells. Okay, and an example of this could be where we're getting all this input from a number of different neurons that do this type of thing, some for seeing, some for hearing, some for smelling, and it produces one specific response, which could be salivation, okay, based on input from multiple different neurons that one neuron is kind of communicating with. The alternative would be diverging circuit, okay, where we have one neuron communicating with multiple neurons, okay? So instead of this previous example, convergence, where we have multiple neurons communicating with one, in this example, we have one neuron that is spreading a signal to multiple neurons. This can intensify the signal. Okay, we can see one axon from this presynaptic neuron sending information to all these postsynaptic ones, and it creates a lot of output and intensified signal. We see this a lot when we are sending commands from the brain to different muscles to help us with balance, posture, and motion. So this might be a good example for that diverging circuit where one neuron is contributing, sending a signal to multiples. All right, moving on to B, we have the physiology of communication. So we're going to take a look at how electrical potentials are changing in neurons, which influences how they communicate with each other. So first, in order to do that, we need to discuss what is present in the cell membrane of these neurons, whether it's pumps or channels. We'll kind of take a look at both, okay? Pumps are defined as membrane proteins, okay? So these are proteins inside the cell membrane that maintain a concentration gradient of ions by moving substances against their concentration gradient. If we're moving against the concentration gradient, this is an active process, meaning it requires energy in order for these pumps to work. And the most common ones that we have on neurons are sodium, potassium pumps, and calcium pumps. Remember, sodium, potassium, and calcium we learned about in the muscle chapter, super important for us to be able to do different types of actions. And it's the same for neurons, where we can pump in sodium, pump out potassium, or pump in calcium, depending on what type of action we want to do. And that's all based off of the chemical synapse by what neurotransmitters are coming in and causing either excitatory or inhibitory actions. So that's pumps, which are present on the cell membrane. We also have a number of channels. Channels are a little bit different. They tend to go with or down their concentration gradients, so they don't require any energy, and typically they work by simple diffusion. We have constantly these channels that we call leak channels or passive channels that are continuously open, allowing different ions to just come into the cell inside and out just by simple diffusion. And then we've already learned about a couple of these when we were looking at the muscle chapter, but we have other channels like chemically gated ones or voltage gated ones that will open or close based on some sort of stimulus. So normally chemically gated ones are closed, but when a neurotransmitter binds a chemical, it can open them. And then normally voltage gated channels are closed, but if we change the membrane potential, that charge on the membrane, then it can open up different channels, okay, or inactivate other ones. So those voltage gated ones are based on if we have a change in the electrical potential of the membrane. For some of our channels, we're just going to go with the sodium ones. If you remember when we were talking about depolarization and hyperpolarization with muscle, and we were looking at, the movement of sodium into the cell and the movement of potassium out of the cell, we're going to kind of take a look here at first sodium and what's going on there with those sodium channels. We're going to call these voltage gated. So once we have a neurotransmitter bind, which is a chemically gated thing, we might change the electrical potential of what's going on at the cell membrane, and that might open up some of these voltage gated channels. So for sodium channels, if we are in a resting state in our neuron, these gates are closed, right? Okay, and they won't open because we're at a resting state. No electrical potential has caused a change in this neuron. And so they stay closed and there's no entry of sodium. Remember, sodium is present in high concentrations extracellularly, so outside of the neurons. We might go into an activation state. Okay, so there's some sort of voltage change across the membrane. And again, we have our channel open. Okay, we would just call it a gate opening. And sodium is able to come into the cell. We know that with sodium flooding into the cell, this is going to change the membrane potential to a net positive or more positive. And then finally, we have an inactivation state. Okay, so we end up having the gate close. The entry of more sodium is prevented. Okay, and this happens for a short time before we then reset and we're back in the resting state. This is what's kind of happening during some sort of activation of these voltage gated sodium channels, where they would go back to being closed once we're no longer doing an action. And this is kind of what it looks like, okay, with these action gates. I wouldn't worry too much right now, the difference between activation and inactivation gates. Just focus on the main actions of these different states. For the sodium channels, resting state, okay, is closed. Activation state is open and we're diffusing sodium into the cell. And then inactivation state is also closed, okay, before we go back to that resting state. You don't have to focus on inactivation versus activation at this point in this course. We have some other ones that we would call modality gated channels, okay? Normally closed, but will open in response to a specific type of sensory stimulus. So instead of a chemical stimulus, like a neurotransmitter binding, these ones will open due to a change in temperature, pressure, or even light, okay? So that's a really interesting specific stimulus. And typically we find these in the membranes of sensory neurons, right, which respond to changes in the environment. One example of this could be the neurons present in your skin, and they might open those channels based on a change in mechanical pressure. This could be stretching of the skin or pushing, putting pressure on the skin. So these modality gated ones are based off of a very, very specific stimulus that occurs to open up those channels. So when we look at a neuron itself, okay, and we're looking at the entire plasma membrane of this neuron, we have a number of different things present. We have leak channels, okay. You can see we have sodium and potassium leak channels. Remember, a leak channel is just always open, allowing diffusion, okay, of potassium or sodium in and out of the cell. We have pumps. Remember, pumps are energetic and they can open and close based on different changes. So we have sodium potassium pumps, either pumping sodium in or pumping potassium out. And then all of these are present here just to maintain a resting membrane potential, right? Just whatever's normal for the cell. So in the neuron, we have a few things going on, okay? And the plasma membrane is going to look different. It's going to have different channels depending on what part of the neuron that you're on, okay? We have different segments is what we would put them into. We have the receptive segment, which is present on the receiving end of the neuron. So this would be the body of the neuron as well as all those dendrite branches. We have the initial segment, which is present at axon hillock. And then we have the conductive and transmissive segments. The conductive segment is the axon and all of its branches or collaterals. And then the transmissive one is right when we get to the end of the axon at the synaptic knobs. And you can kind of see here, we have different types of channels just based on where we're at, okay? You can see that on the receptive segment, and you can see it in this picture here pretty well. We have different voltage gated or chemically gated, excuse me, ion channels, ones that can allow different things in, okay, like sodium and potassium, and even chlorine makes its way in, and chlorine is a negative ion. And all of these things in the receptive segment are part of maintaining our resting membrane potential until we receive some sort of chemical stimulus, which would be a neurotransmitter interacting here. The initial segment is at the axon hillock. There is just voltage-gated sodium channels here and voltage-gated potassium channels. The axon hillock is kind of where all of those electrical potentials culminate. And so we have a lot of incoming sodium and maybe some outgoing potassium. And this is where we might do something called summation, where we get kind of the total of whether we're going to do an action or not do an action. The conductive segment also contains the same channels, voltage-gated sodium and potassium channels. Again, this is whatever happens, whatever net potential we have can cause an action potential or inhibit an action potential. And this depends on what chemically gated channels are receiving, whether they're receiving excitatory or inhibitory neurotransmitters. And then finally, the transmissive segment, we've learned this before, but in order to release neurotransmitters from the synaptic vesicles inside the synaptic knobs, we need to have calcium channels present, and we have both voltage gated calcium channels and calcium pumps in that area. Again, this is just some characteristics. We have a little bit of a different resting potential going on for our neuron at rest. If you remember when we had the muscle cell, a muscle cell at rest is negative 90 millivolts, okay, for that resting potential. It's a bit lower, okay, for the resting potential for a neuron at negative 70 millivolts. And again, we have membrane potential, which is just the charges across this membrane. And again, we're going to be starting at this receptive segment here where we are going to receive a chemical signal in the form of a neurotransmitter, which is going to open and close all these different channels, creating a different charge, okay, where we're going to leave that resting membrane potential and then see if we do an action or not. At resting membrane potential, potassium diffusion is the most important, okay? Potassium we know is in high concentrations inside the cell and it will diffuse out with its concentration gradient, okay, just through simple diffusion. But, okay, this is going to happen with those leak channels, but we might have other things in play like these sodium channels that are open also influencing this because sodium is going to be coming inside the cell. And all of this is to kind of try to maintain this negative 70 millivolts, okay, for our resting membrane potential. So we're going to look at some of these events that occur, okay, at each of these segments. And it might not surprise you that what starts off the events about whether we're going to actually propagate or create an action potential has to do with the receptive segment first with the binding of a neurotransmitter. What we end up creating on the receptive segment is not an action potential itself, but we end up getting all these graded potentials. These are small, short-lived changes in the resting membrane potential because we have different types of neurotransmitters happening, okay, coming and attaching. And it's going to open up different types of chemically gated ion channels. And when that happens, we can either have a bunch of sodium flooding in, we could have chlorine flooding in, we could have potassium flooding out. And we want to see what kind of degree that change is going to happen, because either we're going to do an action potential, which would be depolarization, or we're not going to, because those neurotransmitters are inhibitory, and we would enter a state of hyperpolarization where we even go below the resting membrane potential, meaning we are not going to do an action. So these graded potentials, okay, are happening to the postsynaptic neuron, right? Because we have a presynaptic one sending signals in the form of neurotransmitters. Okay, so we would call these graded potentials, okay, that are happening, postsynaptic potentials. And again, they could have a net excitatory or inhibitory effect based on the neurotransmitter binding. And we could have multiple types of neurotransmitters binding. They could be excitatory ones and inhibitory ones that are binding at the same time. And so we want to get to what is the overall effect. Excitatory ones are going to cause excitatory postsynaptic potential, EPSP, causing depolarization. So we create an action potential, or we can have inhibitory neurotransmitters creating an overall inhibitory postsynaptic potential, which would result in hyperpolarization, where we're not going to do an action potential. And we're actually even going more negative than the resting membrane potential. And I'm just showing this example here. Sodium entering into the cell would lead to depolarization. We've learned this in the previous chapter. And so we would have these excitatory postsynaptic potentials generating. On the flip side, if we have something that is inhibitory, it's either causing the entry of a negative ion like chlorine or the exit of a positive ion like potassium, leading to hyperpolarization, where we become more net negative than our resting membrane potential. And again, this could be happening all at once. You can see in the green here, we have EPSPs, and in the red here, we have IPSPs. And again, all of these are going to culminate together. And depending on what wins out, we'll either do depolarization or we'll do hyperpolarization. And how we add those together is simply through a process called summation. And summation occurs at the axon hillock. Okay, that's our next segment. And again, if you don't remember, that is the initial segment, right? So the receptor segment is getting all of these different signals together, and they all get summed up together in a process called summation at the axon hillock. And if we end up producing, okay, or reaching the threshold membrane potential, then we're going to do an action, okay? And typically the threshold for neurons is negative 55 millivolts. If we do not reach that negative 55 millivolts, we are not going to do an action, okay? Typically, for this, we have what we call an all or nothing law. If the threshold is reached, we do an action potential. If it's not reached, then we don't do an action potential. It is very, very similar. We have this comparison to the firing of a gun. If we have sufficient pressure on the trigger, okay, enough action generated, the gun is fired. Insufficient to pressure, the gun is not fired. It's not it is an all or none thing. It's not, if you have a little bit of this action potential, it's going to hang around and eventually we do an action potential. It's all or none for what we're doing at the initial segment, the axon hillock. And then, of course, we create an action potential. The action potential leads to depolarization, where we have, again, all that sodium flooding in, okay, through those voltage gated sodium channels. And then eventually we will go back down to repolarization, which is when we have the release of potassium, and it's going to cause us to go back down to our initial resting membrane potential. And again, once the action potential occurs, we are going to propagate a nerve signal, right? We're going to be able to communicate with another neuron or an effector. And again, this is just showing that process, okay? And we've seen this picture before. What we have added in here is hyperpolarization, which is something we mentioned before, that if we go even more negative than our initial resting potential, meaning we are absolutely not going to do an action, we might go into hyperpolarization. And again, please review these steps for what different ions are present at each of the stages. This is just explaining it again. You have these slides to help you kind of through each step of the process. Depolarization involves the influx of sodium. Repolarization involves the exit of potassium. And again, if we were doing hyperpolarization, that is something that really occurs at the receptive segment. We might have a huge entry of chlorine or a huge exit of that potassium. In the conductive segment, this is our next part, which is in the axon, okay? We do two different types of conduction, which is just passing that signal, okay, that electrical signal through the axon. Continuous conduction occurs on unmyelinated axons, where we have just kind of this continuous moving of this charge. The other one, which is more common because most of our axons are myelinated, is called saltatory. And this means that action potential occurs only at those neurofibril nodes, those nodes of Ranvier, where the axon's voltage-gated channels are concentrated. And I'll kind of show this process here. What saltatory conduction does is it makes the action potential move very quickly, and the electrical potential kind of jumps from node to node. We can kind of see a picture of that here. This one is continuous. It's going to be a slow signal, but we have, these ions kind of present throughout these channels. This one, we can see kind of the jumping where that signal moves, and it will actually move a lot faster through this saltatory conduction because we have this insulation with the myelin and we're able to move node to node to node. Lastly, we get to the transmissive segment, which is the synaptic knob. And again, if you remember from the muscle chapter, we have calcium that is flooding into, attaching to those synaptic vesicles, and then allowing them to fuse with the membrane of the synaptic knob and release their neurotransmitter. These are our last two slides. I just wanted to talk briefly about myelination. We saw that with myelination of those axons, we can do that saltatory, very quick conduction of that signal. But the myelin itself, depending on its thickness, will actually help with it moving faster down the line as well. Thicker axons, so how thick the myelination is, offers less resistance to the current flow down the axon, meaning we move a lot faster. And then also having these nodes will help that as well. So we can actually classify these into different groups. This is the last slide in this PowerPoint. And these different groups are just describing kind of the diameter of the myelin sheath and how quickly it can conduct an electrical signal. So the group A ones are the fastest by far. They have a very thick diameter and they can move as fast as 150 meters per second. So that's very, very quick. Group B and Group C are a bit slower. Group B is kind of in the middle there, okay? And I don't even have it on this chart, but there is Group Bs in here. They conduct at 15 meters per second, and Group C is very slow. Group C really has no myelination associated with these axons, and they move 1 millimeter per second. So this is just kind of showing, just as an added thing, that not only can we do the saltatory conduction with that myelination where we have it jumping from node to node, but depending on how thick the sheath is for the myelin, it can also make the signal move a lot faster and more straight. Again, Group A being the fastest, Group B being kind of in the middle, and Group C being the slowest because there is no myelin present.